Hello, Varsha there. Is there any participant who has joined? It's okay, Varsha, no problem. As today is the first class, I'll wait for a couple of minutes to have few more people and then we can begin with our proceedings. Okay, so welcome everyone. I am Shrishti Banerjee. I am from IIT Hyderabad, a PhD scholar. So I'm one of the TAs of this course. A warm welcome to all of you to this course. The And so in we'll be conducting two live sessions from each week, and we'll be discussing some sample questions and assignment questions with the idea that you'll be benefited from it and it will help you to solve your own assignments. The questions will be related to assignments, but not the direct questions from the assignment. So how are we going to proceed? There'll be 10 to 15 questions per day, and first we'll be discussing like 5 questions. I'll take a break for 5 minutes, then we can have a discussion among each others if it's clear or not and if we need to rediscuss. And at any point of time while discussing any question, if you feel like you're not understanding or you need more clarity on it, do stop me and we can again go back and discuss that. So that's how we proceed. And definitely use your reaction button to raise your hands or your hand gestures. And definitely you can post anything else if you require some assistance in your chat box. So after like 5 questions, I'll go to the chat box and I'll see what questions we have. And one more thing, uh, yes, and while we're discussing the questions, I'll flash each question and I want all of you to respond the answers in the chat box so that we are able to see that all the answers are at par or not. And once that is done, we'll proceed with the explanation. Okay, so let's begin. Just clarify me one thing. Am I visible and is my voice audible or I need to speak louder?
Yes, ma'am. You're both audible and visible.
Okay. Just also confirm me. I'll start sharing my slides if my slides are moving or not. Okay. So are my slides changing? Anyone?
Yes, ma'am.
Yes, ma'am.
Okay, okay. Thank you so much. Thank you so much. Okay. So mostly we'll be talking about the topics by biology for engineers. That is the first week that you've been have gone through, and I hope that all of you have gone through the contents. So some questions related to that topic. Okay. The very first question that suppose you have been assigned. Just a second. Yeah. And is the recorder still on? Anyone?
Yeah, I
Yes,
think the
ma'am.
recording is on.
On now. So if it is, it kind gets like off, please do let me know also. Okay.
Yes, ma'am. Will
First
do.
day we might have some technical glitches, so just pardon me for that. So just assist me in all that. Okay. Thank you. Okay. Let's discuss the first question. So you have been assigned to a microbiology lab and handed a glass slide to which to investigate the phenotyping of the organism put on it, like what it displays, like when you're observing a microorganism, how it looks like. So after gram staining, you must classify the organism based on the structure seen under 40x microscope and what could be the best possible prediction for the microorganisms mounted on the slide based on its morphology. So your options are budding yeast. Like after seeing this, what do you think? What kind of organism is this based on gram stain? So these are your four options. I would like to have like all of you just reply on the chat that what could be the possible answer for this. I hope the question is clear. Okay, we have one answer from Anika. We have one more answer. Okay, so we have four answers, all D. So any one of you, do you mind to like explain why it's D and not like the others?
Ma'am, like it's gram negative since the staining is pink, and we know that Pseudomonas is actually like a bacilli in shape. So
Hmm.
since it's like coccus, so it must be Neisseria.
Good. Very good. So yeah, that is the answer. I would go through the explanation once again. That is, if we go, I just want to just give an idea that what the gram staining is majorly. We use a particular stain which is known as gram iodine to stain organisms. It's a process of differential staining so that you're able to see the morphological differences between two kinds of different kinds of bacteria. So we try to stain it. Gram positive organisms are likely to take up the stain and turn slightly purplish, whereas the ones which cannot take up the stain, they turn pink. And this mechanism occurs because of its cell wall structure. That's the reason why this difference is seen. And so definitely the organism that has been given in the question, it is pink. So it's a gram negative organism. But now you have two more options that for gram negative, it could be a proteobacter, it could be a diplococci. Now we need to understand what a proteobacter is or a diplococci is. By diplococci, cocci means spherical shaped microorganisms, like somewhere like this. So if we get into the details of that, these are the different shapes of microorganisms. So we see that these are mostly cocci and these are also cocci. So anything which is spherical in shape, that is a coccus. So yes, your answer, you all are absolutely correct here. It's a gram-negative diplococci, preferably Neisseria. Okay, so we'll proceed with the next question. Yeah. So the next question is, I guess it's visible to all of us, till I'll read it out, that biomimetics is the study of biological systems, unique structure and functions, to modeling and engineering of materials and machinery. Pick out such evidences of biomimetics from the following options. So you have three options. So try to solve it like which of the two options could be a classical example of biomimetics. We have one answer from Anika. So, Anika, can you just elaborate why A and C are not both A, B, and C? Like why B cannot be thought of a biomimetics where we are just exactly replicating the activity of insulin production in an eukaryotic system to like a bacterial system? Why cannot it be considered as that?
I think insulin because it's because it says it's a study of the unique structure and functions and to model it all over again. However, the central dogma of the gene to mRNA to protein, even if it's a protein that is not normally produced in bacterial cells, remains the same throughout like eukaryotes and prokaryotes. So it's not taking inspiration from anything, it's simply inserting something new into its machinery. So I don't think we would be over here.
Yeah, that's an absolutely correct answer. That if we look into the three options, option A and C, yes, the answer is like yes, the answer is A and C because if you see, biomimetics is mostly structural and functional modeling of biological things, and that is like interfacing it with engineering, like materials and machinery. By the if we go by the definition, exactly. So, but the thing is, in case of insulin producing bacteria, what happens is that human insulin gene is inserted or cloned within the bacteria to have a higher copy number of insulin and downstream get the enzyme, right? The protein. So it's not exactly structural in that sense, but option A and C, it's like a building or a climbing material that is uh, identically copied from the biological system to make another system which will use it as the same purpose. So yes, the answer here is A and C. Let's move to the, we have one more chat. Okay. So let's move to the next question. So the Human Proteome Project, this is the question. It's a quite a long question. I guess you guys could just have a read and then we can discuss like what were the major goals of HUPO. It was a global initiative by the Human Proteome Organization that intends to change the understanding of the human proteome. So just go through the options and let's like see that what could be the answer. And you can start posting your answers. Yes?
Isn't this question included in the assignment? Because I remember
Yeah. It could
looking
be.
it
It could
up.
be. Yeah, it could be.
All right.
Hmm. Okay. So yes, you just started putting in your answers, and the answer is yeah, it's B. The thing is that it's very straightforward. It's a bit informative question that if we look into the third option, that why third option doesn't fall within this categories. That is, it's mostly it says the project has one major aim to cover the information about the human epigenome, which was completed in April 2003. But here we are actually talking about the Human Proteome Organization. So yes, that particular option doesn't fall within the scope of the question. So yes, it's option B, which is the and options were that it's a collaborative effort, including numerous research labs throughout the world. Like at any end of the world, people were working for it. Second is its goal is to map the whole human proteome in a systematic manner, utilizing both existing and new approaches. And fourthly, it aims to discover the missing proteome of the human body and creates the groundwork for the development of diagnostic, prognostic, and therapeutic and preventive medicinal applications. So, yes, that's the thing. And so, let's go to the next question. So, yes, now we have a slightly experimental question. So, you all guys can have a read of the question and then we can discuss the answer. And if you have any problem in understanding the question, do let me know.
Ma'am, I'm unaware of some of the techniques
Yeah.
mentioned over here.
Yes,
So
yes,
if
yes.
you can explain them.
Sure. We will be discussing that. Anika, just to know which techniques are you unaware of here? Just to know that.
Um, Immunoprecipitation is just adding the antibody to the given antigen, right?
Mhm.
So that I'm aware of. Okay, well, fair enough. Um, I'm just a little confused because it tended to be a lot of technical language all at once. So
Yeah.
if you can
So
explain
actually,
it.
in this options, if you see, immunoprecipitation, mass spec, then protein sequencing, I guess, and there's one more fluorophore staining, conjugate like fluorophore staining. So there are like four processes, right? So I'll be briefing
Mhm.
about all the four, and then we'll decide that which sounds to be a better process of solving this question
Okay.
at the approach. Okay. So, what does the question say? That we are pursuing research on SARS-CoV, and we understand that how SARS-CoV enters into the body. So from literature search, we know that there's a human receptor, ACE2 receptors, to which the SARS-CoV virus binds and it enters. But there are even other cellular proteins that are associated with the ACE2 receptor. So it's not just one protein, but multiple proteins which are associated with ACE2 protein that helps the virus to enter the human cells. So how can we understand, or how can we possibly find a way of knowing which proteins, or identifying which proteins are associated with the ACE2 receptor also? So mostly, but the question, if this question was given to me, I would understand that yes, it's mostly about associative proteins, like interacting proteins, and how to find them, like which proteins are interacting with a specific protein. So from the
It's about
options,
trying to find which proteins are interacting with this ACE2 protein over here.
yes, like what could be the best
Okay.
possible method of characterizing those associated proteins. So
All right, understood.
yes. So where are these associated proteins? It is associated with the ACE2. Like I am not sure of your background, but like whenever we're talking about cellular and molecular biology, it's not just one protein and one receptor, but there are underlying many proteins associated with it that aid a particular function. Okay, Anika. So now we need to see that which method could be the best one. So the first method is like immunoprecipitation experiment with the anti-ACE antibody followed by mass spec to identify the missing proteins. Okay. Then the second option is to completely sequence SARS-CoV, and match its genome with the human genomes to find counterparts of the protein coding genes. Like if we have same kind of proteins that the SARS-CoV have, so it is expected that it will be the same one. Like if the human genome and the genome of SARS-CoV have same kind of proteins through sequencing. And are you aware of sequencing?
Yeah, you're trying to document all of the given base pairs,
Exactly.
right?
Like what kinds of proteins or genes that you have? Like basically, you have a gene, you have your protein, you send it to a laboratory. There's a protein sequencer or like a sequencing instrument, gene sequencer, and you sequence what is the composition of the constituents of your sample. Okay, that is how you do. So that is what is explained in part B. And the third is that you perform a fluorescence staining by tagging a fluorophore conjugated to ACE2 in a particular cell line to identify all associated proteins. Like what happens is that a fluorophore is a compound that will fluorose when under your under UV light. Okay, so if you're conjugating such a tag with a particular protein and you're passing it through a specific wavelength, then it will fluorose and we'll get an idea that okay, these proteins are present. Like if you know which protein you are tagging. And why is a cell line mentioned here? Because this entire thing happens in a cell line. It has to happen within a cell line. So that is a cell. You know what a cell line is? Any one of you?
No, ma'am.
Okay, fine. Then we
No,
discuss.
it's like a certain, like a strain of cells that are
Exactly.
typically
Yes,
used
yes,
for
yes.
laboratory experiments or performances, right? Like there are cancer cell lines available
Mm-hmm.
and stuff like that.
Exactly that suppose we cannot conduct all experiments in human or in model organisms like mice, Drosophila. We cannot do that. We need to see what is happening at the cellular level in the lab. So what we do is we take a part of the tissue and we culture the cells from it, and it forms a cell line. Like for generations, if you grow it, you have a cell line, and in that you are manipulating all your experiments. So you're creating the real environment within the lab. It's a kind of you're mimicking the environment that happens in reality to just conduct your experiments in very layman language, if we have to talk about. So what you're doing is you're conjugating fluorophore tag with ACE, and you're seeing what is happening, and you're then fluorescence, then you're seeing that what is actually happening. So generally what we do is there are many ways of doing this. So we use two different tags to two different proteins, and if these tags together they're fluorescing, then we say that okay, they are interacting. But if they're not fluorescing together, then they're not interacting. So similar kind of approach is mentioned here. And the third one is you can perform a combination of B and C to get the best result. Like you could perform a sequencing, and also you could like do a fluorophore staining to find it. So if you guys have the basic, like what I've said, if it's slightly clear to you, what is your take? Like what process should be like assume here to conduct the experiment? Any idea?
Ma'am, I feel like C would be a more appropriate option.
Why?
Ma'am, like it makes a lot of sense to think that just having a fluorescing particle to attach with different types of proteins will give us like how you said it will give us idea about how it will interact with the other molecules. Uh, I'm not inclined towards B because I don't get a complete visualization of how can we use it to identify the proteins. So
Mhm.
I'm inclined towards C.
Okay. So as you said that like what you understood from my words, that it's easy to understand which proteins are there, right? But don't you think that just having an idea that what proteins are there will make sense, like until unless we are able to identify exactly which proteins are there, that is also very important. You will definitely get some results, like obviously some two proteins will fluoresce and you'll note that these are interacting partners. But to get the exact idea which protein is there, don't you think that will be much more substantiating?
Yes, ma'am.
Right? So everybody is on the same page?
Yes, ma'am.
Yeah. So exactly. So that answer will be A. Perform an immunoprecipitation experiment with anti-ACE antibody followed by mass spec characterization to identify the missing proteins. Now we'll discuss why. Now I have tried to elaborate this technique. Just tell me one thing. Is another box visible in the screen, like which shows my picture and your names?
No, ma'am.
It's not now, so it's happening only for my system. I'm getting confused that you guys are able to like visualize the entire screen. Okay, so uh, what do we do here? It's a very basic idea. Like we can attach the reference, so you can just go back and see what the technique is all about. So if we just think what is an immunoprecipitation and then followed by a mass spec. So let me just first briefly explain these two things separately. So immunoprecipitation is like you're trying to precipitate your target with the help of immune system. Like with immune system, I mean like antibodies. Okay, you have your target, you provide it with your anti-target, like antibody of that, and then it will precipitate, and then you elute or you just take your targets out of it from the entire pool of samples and you do a mass spec. So what is a mass spec? A mass spec is a very high-end instrument which identifies which okay so okay so which identifies that which exactly proteins are present in your sample, right? So for that, you need to take, like, this is your pool of samples. Suppose it could be blood, it could be your pool of samples, and you attach beads which are covered with antibody specific antibodies. Okay. So here is your samples and your beads which has antibodies to it. The beads, like you see, these are the antibodies and these are the beads. They will bind to the protein of interest. Now, antibodies are very specific. They bind to the protein of interest. And then you wash it and you separate only the beads that have been bound. So that beads have antibodies and they are bound to the protein. So in this solution, you just have your target proteins, the beads and the antibodies, and then you remove the beads and the antibodies through a process called elution, and you have your samples, and then you go for like mass spec, which will tell you that exactly which proteins you have or the which associated proteins you have. So what do you think, that this is a better technique of doing it, or still you guys are inclined towards C? Not because of the fact that I am saying that this is the correct answer. What do you guys in general think?
Ma'am, I don't know the like capabilities of mass spec. Like I don't know. From my understanding of it, isn't it normally used only to identify like organic compounds? I mean, I
No.
suppose proteins are organic compounds.
Yeah, yeah.
So
Like,
like
hmm,
but the only thing is I don't understand the technique of mass spectrometry that well. So I'm more inclined toward the technique that I was able to understand better.
okay, okay, okay.
So I cannot like say which
But
is more
I can just tell
accurate.
you that if you are using, like, if you go back and just plain and simple basic research on mass spec, you'll understand that it's a wide range of sensitive. It's a very sensitive instrument. It can like identify millions and thousands of proteins together. So when we are talking about identifying and quantifying proteins, then mass spec comes before any technique from a
Okay,
unknown
then
sample.
definitely I can understand why A would be the better option.
Yeah, because you're
Because
actually merging two very important techniques. First, you're precipitating all your proteins that you have, and then what you're doing, then you're sending it for mass spec analysis, the proteins that you've got from this precipitation experiment. So
also, I think it would reduce the human error because most of the
definitely,
computation is taking
definitely,
place by the
definitely.
instrument itself.
Though
So,
though there is a chance of human error, like no process is foolproof as we know, but yes, definitely, definitely. So okay, so
Ma'am,
we are done.
like,
Yes, sure, sure.
I have a doubt in this question, ma'am. Like, when we
Niranjana,
refer that
just
there are
just I would like to interrupt you, all of you, that the time, there's a time limitation of one session. So all of you need to rejoin the session, okay? After like 2 or 3 minutes, when 2 minutes are left, just leave the session and rejoin it again. Okay.
sure, ma'am.
Yeah, you
Ma'am,
can
like,
just tell your question. Yeah.
I'm like they have mentioned here that there are other cellular proteins that are associated with ACE2 protein and help in virus internalization. So, are these cellular proteins like the ones on the cell wall or the ones that are present inside and are involved in the cell functioning?
It could be both. It could be both because
So,
when we are talking
like
about
how?
viral internalization, so there's a receptor, the viral particle will bind to it and then it will be internalized. So some could be exactly from the, if we look from within the cell, some could be exactly like very in the close vicinity of the cell wall, and some could be slightly away from it, and then it could be like interacting partners to it.
So basically, these need not be the only ones that are allowing the virus to enter the cell. They might be the ones who are like converting the viral DNA into RNA and all such stuff.
Yeah, could be. It could have multiple properties that we need to look into. That is why we need mass spec, no? Because from mass spec we'll understand that what is the sequence, what is the peptide composition, even what protein it is. And if it is in the database, then you will also understand that which proteins are there. And if it's known proteins, then it's again a very like substantiating data. Like there was a very good paper by Gordon et al. During the COVID times that they identified like many human interactor proteins that we have, like human counterparts that are interacting with the SARS-CoV virus, like proteins. So that's the beauty of this technique, that you get to know a lot of about the details and very clarified. So yes, that's important. So I'll just leave this meeting for 5 minutes and then we'll rejoin. Okay. Because four minutes are left and then we can go on to the next question, so you guys can rejoin immediately. Okay, is that clear? Let's just wait for a while for others to join and then we'll begin. Yeah. Let's begin, okay? And we can like take up from where we stopped. We were in this question, right? So Niranjana was asking me a question, right? That only is it only one protein or like multiple proteins and not necessarily the cell wall or the cell membrane or the or internal proteins, right? So are you clear with the question? Answer?
Yes, ma'am.
Okay. So let's begin the next one. Hmm. Okay. So a very straightforward question. This one again. So it says that a ribosome is a complex molecular machine found inside the living cells. They're made up of few basic biomolecules and perform one of the most crucial functions for cell to live. Point out the correct functional and structural information for this organ. So options A, B, C, and D. So what? Just give it a guess what it could be. So yeah, answers are correct. So can any one of you just explain, maybe Swayam, that why it's just a second. Let me take. Um, Yeah, like why is it C and not the rest?
Basically, the ribosome reads the messenger RNA, the sequence on it, and translate that genetic code into a specific string of amino acids. Like it create a chain that it create a chain of proteins or something like that.
Hmm. Yes. So it's actually has a dual function, right? That it can convert the encoded signals. Like I'll just read out what is in the option that it is in charge of converting encoded signals from mRNA molecules into amino acids. So the ribosome is actually made up of both RNA and protein. It has both, like it has both ribosomal proteins and also the ribosomal RNAs. If we understand the structure of it. So yes, it's C. Structural basis, it's C. Now, yes. So as discussed, 5 questions are done, and now maybe we can like have a discussion if you have questions from other like if you have any other questions related to the session. Otherwise, we'll carry on with the question answers. So if you want some other concepts or some questions that you need further discussion or any topic, and otherwise we'll continue. Anything.
No, ma'am. As of now, from the material, I was able to understand everything as well as the extra material, like the extra reading that was given on the Telegram group. I am yet to go through it. So, if there's anything with that, then I will like get back to you at the next live session, definitely.
Sure, sure. And you can keep posting on the Telegram channel like your doubts and so that we can we like both the TAs we can prepare for it and we can address your questions like both of us.
All right. Okay. Thank you, ma'am.
Yes, yes, sure. So then we'll proceed with the next questions, I guess.
Yes, ma'am.
Yeah. Okay. So again, a picture diagram. To we need to identify which organelles are these in the correct order. So A, B, C, and D. Swim C or B? B. Okay. Anika disagrees with the rest. She says it's D. Anika, why D and not B?
Oh,
As
my
well.
bad. No, I meant to type B. It was an accident.
Okay, okay. That's fine. That's fine. Yes, so the correct answer is, I guess, B. So it's very visible. Like uh, the outer layer is mostly the cell wall, but within that we have the cell membrane. So A is cell membrane, B is the chromosome, C is the plasmid, which is the extra chromosomal DNA that we discuss or we know, and D is a reproductive part. So yes, that was correct one and easy. Okay. So the seventh question. Now just have a look at the question and then we can maybe discuss that. What is it? Or I'll explain that's better. That you're working in a laboratory where you're instructed to culture an unknown strain. So you're given an unknown strain and you're asked to culture it. After 48 hours of streaking on a culture plate. So are you aware of the technique called streaking?
Yes, ma'am. To isolate bacterial or microbial colonies, we have to use streaking because otherwise the number of colonies is too high for us to study it
Yes.
properly.
Yes. So how do you use it? Like what is the instrument or the apparatus that is required in streaking? What do you call it?
Inoculation loop, or you can use a needle, either depending on what your thing
Yeah,
is.
good. So you have done it in hand, like streaking?
Yes, ma'am. We were taught in the first semester, so.
Good, good. That's good. Great. So you given and you have just opted for streaking in a culture plate, and you observe that the bacteria is having a extended thread-like morphology. And if you see the picture like this, this is the extended thread-like morphology. So what is this organelle? Like what this organelle could be called, and what is the functional structure, and how it benefits this bacteria from it? So okay, answers. Okay, see. Okay. So what is the function of flagella, if anyone? Swayam or Anika?
Ma'am,
Locomotion.
Good. I guess there's a new one, Smriti. Yes. You can also
Yes, ma'am.
participate. Yes. Locomotion. What kind of locomotion, Smriti? If you're saying it's like C, then you're specifying the function as a rotatory movement. So
Rotatory. Yes, ma'am.
rotatory movement. So it'll propel the bacteria to move forward or backward, right?
Yes, ma'am.
Okay, okay. So yes, you're correct. It's option C. The organelle is quite like visible. It's like quite clear. It's a flagella, and the function is a rotatory movement. So it rotates by producing ATP. Like it has a role with ATP production also, and it helps the bacteria to move forward. Okay. That's all for this question. Then let's move on to the next question. Nice. So here's a question that intracellular signaling happens in variety of ways inside a human body. Following is a diagram representing which type of signaling. You have four different options. Have a look in the diagram and just guess what kind of signaling is it. Suppose this is the signal molecule. It is going inside the cell, again coming outside, or it's a signaling molecule which is coming from the cell and again it is inducing something. So what kind of signaling process it could be. Okay. Niranjana and Swam says it's 3. Anika is more inclined towards 4 and Smriti also 3. So okay. Anika, you need to defend why it's 1 and 1 against 3. So yes, Anika.
I could be wrong, but
No, no,
according to my understanding, paracrine signaling is when it's across like short distances. However, autocrine signaling is when it's like the same cell is the receptor as well as like the 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 signaling cell and the receiving cell are the same. Like so, as we can see over here, that the signal molecule is like originating and getting like detected by the same cell itself. So, according to me, it should be autocrine.
okay. And the rest who are more inclined towards paracrine. So what is your understanding on paracrine signaling? Maybe Smriti, Niranjana, Spam, any one of you.
Ma'am, like as we can see, the molecules are like discharged into the extracellular fluid, and only then they are reaching the nearby cells. So I thought it must be paracrine
But
signaling.
it's actually excreted from the same cell, right? It's again like excreted or like yeah, excreted from the same cell, and it's again going inside the same cell. Like it's a cycle
Then
happening.
it may be autocrine, ma'am.
It may be autocrine, right? And what about Niranjana?
Yes, ma'am. Now I think it might be autocrine.
Autocrine. And one more was then Swam, I guess. Yes.
No,
Swam,
I agree.
you agree to it.
Autocorrect.
Okay, let's do something. Let's see what are the different kinds of signaling in picture, and then maybe we can come to a conclusion that what could be the answer. Let's have a look at it first. Okay. So, see, there are 4 different types of signaling that is happening. Okay. One is contact dependent, that it happens like a signaling molecule. Membrane bound signal is there and it goes and binds to a receptor on the target cell. And then the downstream process is happening. It could be paracrine. For paracrine, if we see that this is a signaling cell from which different like molecules are being excreted and it goes and bind to target cell which is not at the vicinity but slightly proximal to it. Okay. And there's one kind on the synaptic, it happens exactly in this within the synapse, like target cell neurotransmitters. So we can just kindly, like currently we can just avoid that for the time being. And there's endocrine. So with endocrine, what happens that hormones, which are transported through blood and then they go and bind to the target cell. So if these are the different kinds of signaling, so definitely it won't be paracrine because paracrine happens like the target cell and the excretory cell is the different. The secretory cell, I'm sorry, it should not be excretory, it should be secretory. The signaling cell which secretes the signal into the outside, that particular cell and the target cell is different for paracrine. And for endocrine, it's the bloodstream through which it is transported. Contact dependent, we've seen that a particular entire signal bound molecule binds to the target cell. And rest is the autocrine. So here the picture is not there, and that is why I kept this picture on purpose. That autocrine, what happens is the cell itself is a target cell as well as a signaling cell. It secretes its own signal and it is again activated by it itself and it accepts it, it binds to the receptors, and this process goes on and goes. So the same cell is both the signaling cell as well as the target cell. So
What is an example of this kind of signaling, maybe?
I have to look into that. Like right now, I'm not remembering, but I'll get
Okay,
back to
okay.
it in the next slide. Okay,
All
autocrine
right.
signaling. Like Mostly in case of quorum sensing, if do you have an idea of what is quorum sensing?
Quorum sensing.
Yeah. Have you heard of it?
Uh, no.
Okay, okay. So which background are you from, by the way?
I am in my first year of BTech Biotech, so I've
Okay,
done like PCBM.
PCBM. Okay, okay, okay. So from BTech background. Uh, so in quorum sensing, then quorum sensing will be difficult for you. So basically, you will have some specific cells, okay, and they are going to secrete some signaling molecules and it's going to act on it. So are you aware of the immune system that macrophages,
Yeah,
IL-6,
yeah, yeah.
IL-7? So
Yes,
suppose
yes.
the macrophage is like, suppose now it actually happens, the macrophage secretes IL-6 and that
Okay.
again activates the macrophage.
Okay, all right. Got
That
it.
is how it's happening. So you'll find plenty of examples. Just go back and see what more examples you can get. And we can have maybe have a discussion next day again about autocrine signaling. Okay.
All right, definitely. Okay.
This is one of the examples that I'm able to recall right now. So yeah. Okay. So yes, we have this question. Oh, okay, autocrine. Right. Okay. The ninth question, the penultimate question is we have to pick up the incorrect statement out of it about eubacteria. So what are the guesses? A. Okay, so Anika is in their team now. And Swayam, what about him? Okay, so I'll just discuss. Maybe A. So are you aware of the four options? Like, what are these? Because I guess you're not.
Um, yeah, it's like part of the Six Kingdom classification,
Ah,
right?
so you all are saying like A, B, and D, the options A, B, and D are like quite related, that they are related to
Yes, ma'am.
extremophils or eubacteria. Okay. But C is, goes slightly different. Like if eubacteria is related to bacteria, then why not F-met? F-met is its initiation. Like we know that, you know what's translation? There's an
Yeah,
initiation.
Yes,
that's
ma'am.
the
Yeah,
central
Yes.
document.
yeah, yeah. So there's an initiation codon, and after that, that codon is AUG, and before that, F-met is like F-met is the initiator amino acid, if we say in that. But it F-met is present in bacteria and normal methionine in case of eukaryotes, if I'm not very wrong. So
Yeah,
if
yeah.
eubacteria is close to true bacteria, yes, sorry.
Ma'am, because if in eubacteria is sort of like I thought you, so like you from
Eukaryotes.
eukaryotes, it's a
Okay,
bacteria,
okay,
so it can't
okay,
be methionine.
okay, no.
So it might be formal methionine.
No, 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 no. Nothing like that, but it's very close to bacteria. Eukaryotes are quite different from bacteria. Eubacteria is still closer to bacteria. It's like within archaea and bacteria. Okay. If you know the three kingdom classification and how it happened, the sixth kingdom and all. So it will be actually option C is incorrect here. We'll see if I am correct or not. My take is no. C is also fine. That's what. Which one is incorrect? That it's a much more evolved form of bacteria and very close to archaebacteria. It is not evolved form of bacteria. It has come before bacteria. So
Oh, so bacteria evolved from eubacteria then.
yes. And then there's another line of evolution which was archae. So you all were like A is incorrect. Eubacteria are extremophiles surviving in extreme environments like hot spring and a deep sea. Listen, whenever these kind of questions come, my approach towards solving is that I try to understand the options. Like whenever we're talking about bacteria, now we know that how versatile they are. Like they can survive in extreme salt condition, they can survive in dry conditions, in heat, in cold, they can be anywhere. So anything which is related to bacteria, put your first guess that no, this is related to bacteria and like obviously you are in a very novice phase of your career, so you might not be having that clarity, but generally eubacteria and these kind of questions, they are extremophiles, they generally, okay, and they're also known as true bacteria as we've discussed, and their initiation process, translation process, synthesis process is fmet, like all of them have fmet, which is initiated by this amino acid, and in case of eukaryotes, it's going to be just methionine. So the D is option is incorrect. That evolved much, is a much more evolved form of bacteria and very close to archaebacteria. So that's where we
But
stand.
the only reason I think all of us picked A was because we always say that okay, archaebacteria are extremophiles, right? So we
Yeah.
just like assume that okay, that might
I
be the
understood.
wrong
I understood.
one.
That's fine.
Yes,
But
ma'am. Same logic.
if you read more about bacteria and like if you see the different habitats or conditions, there are thermophiles, there are extremothermophiles, there are lyophiles like this, that they have different habitats, and bacteria are also present there. Like you'll find bacteria in extreme, like in the coming classes when you guys have actually gone through week 2, right? The PCR and all of these PCR techniques and
Yes, ma'am, we have
uh,
gone through it.
just I'm not discussing anything about it. So in PCR, the polymerase that is used is a Taq polymerase, generally used. So that is actually obtained uh, from a bacteria which survives in a very high temperature conditions from some thermal vents.
Thermus
Thermos aquatic.
aquaticus.
Yes.
Thermopsychotic.
Yes,
Yes.
yes, exactly. So you can have an idea na, if bacteria is surviving in that. So these are basically extremophiles, like thermophiles. So extremothermophiles are the one extreme temperature. So like this, there are different categories that we can discuss if we have more like opportunities in the future. So yes, it will be D. Okay, clear? We'll move on to the next question.
Yes, ma'am.
Okay, okay. So the last question is, mature cells in plants are considered to be totipotent. Choose the best choice from the list below. So, which according to you is the correct answer? Should cell images. A, maybe A, D, I think A. Okay. So, what is your understanding about totipotent cells? What do you understand when somebody says it's a totipotent cell? Anyone?
Single
That
cell can differentiate into many
Okay, okay.
different type of cells.
A single cell
Can differentiate
Can
into many
giving
different type of
rise to an embryo or
embryo. Cells giving rise to embryo. No, no, need not like stop. You can just clarify. Like we are having a open discussion here. Don't be scared.
Ma'am, I think totipotent cells are like embryonic stem cells, so they are not able to give rise to an entire organism. However, they can
blast can give
differentiate.
rise to a blastomere.
Huh? You're right. You were actually going right, Smriti. You just stopped
Yes, ma'am.
saying. You were actually cells giving rise to embryo, like a complete embryo. So what is an
Yes,
embryo?
ma'am. Complete
It's formed of many different cells, right? So you were right
cells.
in
Yes.
that. You were right actually. And Anika, you're saying something?
Yeah, just that it can't give rise to an entire organism. However, it can differentiate into many different kinds of cells.
Good. So basically, these are cells of a particular lineage that can differentiate and form new different kind of cells. So before getting into the example of plants, I would just come back to a very classic example of totipotent cells present in humans. Any idea? Any idea? Like the best totipotent cells we have in our
Stem
body?
cell.
liver.
Embryonic stem cells
Liver
are the
cells.
ones that you get from
Liver
the
cells,
liver
actually.
cells.
Liver
Okay.
cell. Yes.
Hmm.
Liver cell.
That's the best thing that's how I remember how what is totipotency. Like we see now during liver surgeries and liver transplants, many a times they're not able to transplant, they cut off a part of liver and then leave it like this. So it regrows back. Why? Because it's a totipotent cell. A liver is the only organ that is capable of regrowing back is liver. So that is a classic example of totipotency. So if we keep into mind the characteristics of okay, same things. Okay. So if we keep in mind of
Excuse me, ma'am, I had a doubt.
yeah, sure.
Ma'am, totipotent cells divide into a specialized cell and they make a whole organ, and those organ can build a
Yes,
whole organism, like something like that.
yes, they can. They're capable of doing that. Yes, that's what happens. Like uh, look, if we say something has a stem cell like property, what does a stem cell do? It originates or give rise to new cells, new kinds of cells. And then it follows a particular lineage. Like it could be epithelial cell, it could be, like, if I say it in a very easy, it could be a lung cell, it could be kidney cell. It's not very scientifically correct of saying like a lung cell or a kidney cell, but just imagine that each different type of cells. Okay, that could happen from one stem cell is capable, like that one stem cell can become a liver also, it can become a kidney also, or it can become a heart. So that is the capability of a stem cell. So what happens is it differentiates to form it. And exactly a totipotent cell, what is the difference between a totipotent cell and stem cell? It has stem cell like properties, that it can also give rise to or re-rise to a different type of specialized type of cells. So ma'am, am I clear in this?
Yes, ma'am.
Yes. And so if you're giving rise to new cells, it can aggregate and form a different, a complete different organ also. Like Smriti said, a complete embryo. Definitely that can happen. It should be an embryonic cell and then it differentiates and form a proper, like a organ. So yes, the answer is A. It possesses stem cell-like properties and can de-differentiate into all of the specialized cell types. And why not B and C? B and C is like ability to differentiate into downstream cell lineages only. By downstream cell lineages means that if particular cell is of a particular lineage, it will differentiate and form that lineage only. But no, a totipotent cell can be any cell and then it can form any other different kind of cell. And they also help transforming neighborhood cells more. This is a specific characteristic of cancer cells that is not happening in case of totipotents. So yes, we are done with 10 questions. So yes, I would like to, all these things will be shared with you. Okay, don't worry. We'll be sharing. Yes. Yes, we'll be sharing all the contents, like the live session recordings have been done. It will be uploaded in my YouTube channel, and also we'll be sharing the slides and with the possible explanations where it has been given. So these will be shared with you shortly. And I would request all of you that because it's a 2 hour live session, you should also come up with different questions and we all can solve it together. So, and that questions, or you can come up with topics also that you want more discussion on, and maybe we can like discuss that and cover it up in the 2 hours. So, yes. So, do you have any questions? Any of you regarding the both the weeks, if you want to?
Ma'am, there's one question from the assignment of the second week that I'm just not able to figure out.
Hmm.
So if you can clarify that. I
Yeah, yeah,
think
sure.
it's the
On
third
what?
one on my like assignment.
So,
The
I'm not supposed to directly give you the answers, but maybe we can have a hints. But again, in 6 minutes, this meeting will get over. So, wait, where is it? I'll share the assignments with you, the present ones. Okay. Yeah. Assignment 2 you mentioned, right?
yes, ma'am.
Assignment 2, which question? Hmm. Third one. Okay. This one, right? Ramesh has been provided the
Yes, ma'am.
plasmid, this one, 15 kb. He observed the following fragments with different combinations as mentioned below. Okay. Upon cutting the plasmid requirement. So have you just tried drawing it? Have you been taught how to draw it?
Yes, ma'am. We like it's mentioned how to make a plasmid map in the in the lectures.
Hmm.
However, my confusion is that even if I draw it, like how am I supposed to A, put it together, and B, be sure that the sites are in the right place according to what I'm drawing? So then
Okay.
I just sort of blanked and I was like, okay, what?
Okay. Okay. So the right directions can only be understood if you like consider two first. Like suppose you have BAMH1 and STU1 give 9 and 6 kb, right? So again you have EcoR1 and STU that gives you 6 and 9 kb. And the different one is not 1 and EcoR1 that give you 4 and 11 kb. So the thing is this 9 kb could be divided into 4 or the 6 kb could be. So you need to find the commonness between these, like which are the common ones and which directions. Like it shouldn't be that not 1 and EcoR1 are giving you a 4 and 11 kb. That means that it cannot be divided from within, like between that. Okay. So try drawing it and try all the combinations possible. And once you submit the assignment, we can discuss that how exactly to solve it. Okay.
All right, ma'am.
Yeah. So when are you generally asked to submit the questions?
I
Right
think
now
both
the
the
deadline
weeks.
for both the weeks is 8th Feb. So
Okay, 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 okay. Next week, anyway, we'll be discussing on details on all of these, so you'll get a clarity. But I guess today we don't have that much of time. Otherwise, we could go to. So I guess the session seems done, I guess.
yes, ma'am. Next session will definitely try and get some stuff from our side as well, so that we can have
Yeah,
like more
like
of a
you can
discussion.
not just questions, but you can get topics also. Like somebody mentioned that
Yeah, yeah.
he or she did not understand lac operon, I guess. Positive control of lac operon.
Yeah, no, I was unable to understand that in the first like the first time I looked at it. But then when I saw that one of the resources sent was also about the positive regulation, so then I was able to look at it and I understood it a little better.
Okay,
So
okay, then that's absolutely fine. Otherwise, I would have discussed it today if you like. But it's okay,
yeah,
fine.
yeah.
If we like keep on doing it in this way, you also bring something from your side, then it'll be more interactive. And like that's the whole purpose of this live sessions to
Hmm.
discuss it. Okay.
Yes, ma'am. Definitely will do.
Yeah, yeah, yeah. Okay. So okay, that's all for today. So I'll be posting all these things soon, and then we can like see you on next Friday, same time. Okay.
Okay, yes. Thank you, ma'am.
Okay. Okay.
Thank you, ma'am.
Thank you, ma'am.
Thank you. Thank you. Thank you all.